Bonjour, professeur Athanas.
Bonjour Mars,
Tu vas bien?
je vais bien.
Ah, alhamdoulilah.
Et toi?
Tout se passe bien?
Oh, ça va, on s'accroche comme on peut.
Ok. Ok, tu as des nouvelles de notre collègue là, Seydou, Alpha.
Oh non,
Mhm.
non. Euh, Ça fait 2 jours.
Euh,
Hello, everyone joining. Thank you for joining.
Hello.
Hello. Hello.
Hi.
Hi, everybody.
Good morning, everyone.
Hi.
So I think we are at a couple minutes past, and I have limited abilities to see people's. Let's see if I can fix this here. So what we're going to do is, I think you can see. Can you see my screen that has the agenda on it? Yes. Okay.
Yeah.
So we're going to do a really quick intro from a couple of us who have been working together to get this off the ground. And then we're going to go on to the details, and then hopefully have time at the end for any of your questions and answers on the ambition and the details. So I'll go first. My name is Mara Lonizak, and I'm a senior group leader at the Sanger Institute in the UK. And my team works a lot on mosquito population genomics, anopheles mainly. And that is part of what has been motivating us to get involved in starting this Culicity global project. And I will hand over to Miguel to introduce himself.
Hi, everybody. It's a real pleasure to see so many faces. My name is Miguel Garcia Knight. I'm based at the National University of Mexico, the UNAM, in Mexico City. And I'm a Sanger International Fellow working with Mara to realize this project. My interest is in viral transmission and arboviruses. And I'm very interested in comparative genomics to see how mosquito genomes may have evidence of past and current viral infections, and as part of my interest in this genomics project. So I'll talk a bit about some of the work that I've been doing from with our colleagues here in Mexico.
And Bernard?
Okay. Hi, everyone. Nice to see everyone. My name is Bernard. I'm a new PI at Princeton in the EEB department there. And I'm interested generally in biodiversity. We've been doing a similar project for Drosophila. And what brings me kind of to this is that we're seeing a lot of interesting patterns using comparative genomic and population genomic approaches in Drosophila, and it'd be really nice to have systems to compare against that are closely related. So very interested in seeing what happens with mosquitoes.
Great. And then Lindy McBride isn't able to join today, but she's also been one of the four of us who have been putting this together. Also on the call with us, we have a variety of people from Sanger Tree of Life, including one person who will give a presentation, and that's Vasil. Actually, he's not from Tree of Life, he's just more broad in Sanger, but he's been working on Nagoya and ABS, and will present on that shortly. So I'm going to kick off first and go through what's going on at Sanger Tree of Life and how to onboard as a partner in a high level so that you understand what you're signing up for if you decide to participate in the project. So if I play this from start. At the Tree of Life, we're all a bunch of scientists, but we have the ambition to create reference genomes across the Tree of Life. And we do this through a variety of different projects. And I just want to emphasize that we've kind of built what we call the Tree of Life engine, which is really genome engine, which is really each of these little boxes is actually a very skilled team of people who know how to do this sort of at scale now. So we have representation on the call from the Tree of Life samples management team. We have a Tree of Life core lab who does all the DNA and RNA extraction. We then send our tissues, our extracts over to the scientific operations where the long read sequencing and the RNA sequencing and the high C is done. Then it comes back to us in Tree of Life where we have an assembly team who put all the pieces together. And then it goes to the curation team who look at that and make sure that it's actually all good and there's not a bunch of mistakes. And where there are mistakes in the assembly, they fix them. And they also submit those assemblies then to the public database. Then we write a generic note. This is called a genome note. It's not really a scientific publication. It's really just a mark in the sand that says, here's what we know about this sort of specimen that contributed to this genome, and here's the genome stats. And then Ensemble or RefSeq can pick up that genome and all the RNA-seq data associated with it and then annotate it. So just to be clear, as part of Tree of Life, we don't do the annotations. The data the assembly has to be released for the annotations to happen, the gene annotations. So we are core funded by Wellcome at Sanger Institute, and that's enabling us to deliver 3,000 genomes per year. But one of the things that we've struggled to get funding for, long-term sustainable funding for, is the sample collection. And so this project, Culicity Reference Genomes Project, is really an effort to kind of, we're starting to do some of these. There's also one on butterflies in Europe, is to start to think about how can we work across a larger team in which people are collecting anyway for their own work and make some of those specimens viable for reference genome sequencing, initially here, but hopefully as time goes on in many other places. So just to emphasize the skill of the teams we have here, they have been able to put together the mistletoe genome, which is 100 gigs. Each chromosome is 3 times the size of a human genome, but also been able to put together a species that is only 1,000 cells big using amplification approaches. We are involved in a lot of different projects, but all of the projects we're involved in, including this one, the Culicity project, will contribute to the vision of the Earth Biogenome Project. So if you don't know about that, it would be worth looking it up. We've released over 2,500 species genomes so far, and this is the titles of some of the genome notes just randomly selected. And it's across the entire tree of life, not just insects. And we have a lot of different portals where you can go to find information out about the data or the progress or the publications. We also have led on a lot of training. So although probably in this case for the Culicity project, specimens will come here and kind of disappear from your view for a year as the genomes get generated. If you're interested in learning how to do assembly or how to curate, there's a YouTube channel with previous videos, and I suspect the BGA, the Biodiversity Genomics Academy, will run again. Okay, so why are we doing this project now? In my team, we've been doing high-quality reference genomes from single mosquitoes for a number of years, but we've been doing this with Anopheles where there's tiny genomes, 250 meg genomes, and it's still been pretty tricky. It's been right on the cutting edge of kind of like what is possible with the amount of DNA you can get from one mosquito. Recently, in collaboration with Lindy, who's not on the call, we generated a single Culex pipiens genome, which is twice the size. This did require some top up, so we generated the normal long read, but then we had to amplify some leftover material to top that up, and we had a separate individual for high C. And really, 80s has been out of reach for this kind of single mosquito approach until very recently. So very recently, PacBio announced 2 exciting things. One is a 4-fold decrease in the amount of DNA required to do their sort of standard input library. And the other is an amplification-based approach that goes down to a nanogram of DNA for up to 3 gigabases of genome. So this, we have not tried either of these yet, but we believe that these will work. And so this really does open up the possibility, I think, to generate almost single specimen reference genomes for most mosquito species. So that's really kind of what motivated kicking this off now. And also, it will take us all time to get all the specimens collected, et cetera, and technology just keeps improving as the months go by. So this part is really, really, really important for me to get across because I cannot overemphasize how important it is to make sure that the specimens, you know what they are, and that they were alive when you process them. Because that's in your control and not in our control, we've had multiple examples of where specimens have come to us and actually all we've sequenced is bacteria because the specimen probably died before it was preserved or frozen. And the DNA looks like it's going to be great. You get nice high quality DNA from bacteria that have grown on a dead insect. So it's absolutely imperative that the specimens are alive at the time you catch them and the time you process them. And I'll talk about how to process here. Although I'm saying we're aiming for 1 mosquito per genome, the reality is things go wrong. And also we're right, as I said, on that kind of edge of what's possible. So having multiple specimens per species is going to really make this a much more successful project, I think. So we're asking for each of you to submit at least 10 and up to 20 individuals per species, if that's possible. We want to spend our money on genomes, not on shipping. And so we have developed a room temperature approach that we have tested on mosquitoes quite extensively. And we believe we can get just about the same quality and quantity of data that we need out of room temperature shipped mosquitoes. So ideally, when you're collecting, you have access to ultra cold temperatures and keeping them on site at ultra cold temperatures, but the shipping process itself can be done at room temperature overnight or over the course of a week. And what we have tested is 1 week at room temperature in ethanol or 1 week in RNA later or all protect. And these are fine, with the caveat that you need to do a slight squish, and I will show you what that means shortly. The failure rate does go up when things aren't kind of the gold standard, snap frozen and kept in -80 for their entirety. That's largely because we tend to get slightly less DNA, but that's kind of helped by the fact that we're asking for at least 10 individuals from each species. If you happen to collect more than 20 per species, this may be of interest for other projects. So we ask you to keep them at -80 or whatever, using the kind of gold standard preservation. Just put a specimen in the tube, ultra cold temperature, and leave it there. You don't necessarily need the preservatives. The metadata that are associated with those specimens, we do want you to keep in case they end up coming to us later, but we don't need to see that metadata. Okay, so here's how to prepare your specimens for DNA. We ask every tube that comes to us to have a barcode on it, not just a handwritten kind of number one, because we're getting tubes from so many different people. We can't have handwriting and simple labeling. We will send you these barcodes if you don't have them. We are asking for mosquitoes that are going towards DNA to be put in whole. If you can see this picture of a mosquito in the ethanol, just pop it from living into the ethanol. It'll die in the ethanol. And then you just take the pipette tip or this is a purple plastic pestle, and you lightly press it against the wall of the Eppendorf and crack its cuticle. You're not trying to grind it or squish it. You want to be able to pull that mosquito out intact as a whole. So this is post cracking. You can see that there's a little bit of material, a little bit of the innards have come out, but we can still pull that mosquito out in one piece from the preservative. What we have found is that this squishing process really helps the preservative penetrate the mosquito much faster, and the DNA is higher molecular weight, and there's more of it from pretty much every preservative we've tested. So it's an important part of the process. And then for RNA, it's exactly the same thing, except that we're saying use RNA later or all protect. Ethanol does not do a good job of preserving RNA. We will not, as I said, we will not annotate the genomes, but we will generate the RNA-seq data that will be released and will be useful for annotating the genomes. So if for some reason you're unable to collect extra specimens for RNA, we can still generate the reference genome, but we won't be able to give you a good annotation. The annotation will be based on RNA-seq data from other species. Okay, so a couple of other considerations. If you're out there collecting and you actually end up collecting both sexes, we ask that you try to split that evenly across the preservation approaches. So for example, that we don't get all females that are in ethanol and all males that are in all protect. It's good to have both sexes in both preservation approaches. If you have fewer than 10 individuals per species, get in touch with us. We may still want to give it a try, but we just want to caveat that the chances of successful reference genome from that are lower. If you're not doing fresh collections, but you've collected in the past using kind of gold standard approaches, so you snap froze your samples, but it's from like 6 years ago, you have those still in the -80, get in touch. We might be able to generate genomes from that. If you already have extracted DNA, it's unlikely to be high molecular weight, but sometimes it is. However, if you don't have any specimens left from that species, then we won't be able to generate the Hi-C data, because that doesn't take DNA, that takes an organism, because it's going for the nuclei. And then we won't be able to put together chromosomes without Hi-C data. So we really need specimens, not DNA. Once you've collected, we want you to keep everything in the coldest temperature you have available. If you have a -80, please keep everything always in -80, so that the only time they're ever at room temperature is when they're being shipped. If you don't have a -80, just store it at the coldest temperature you have available. As I said, we want to ship everything at room temperature, typically overnight. If you're able to pay for that shipment, that would be great. And if not, we're likely able to reverse charge the shipping costs. We will not accept any specimens until all of the paperwork and processing is complete. So anything that arrives here without that would end up being likely destroyed. So please don't ship anything until you've been asked to ship. Also, we've had cases in the past where things have been shipped without the proper paperwork accompanying the shipment, and we send that paperwork to you when we're ready to receive the shipment. So the import process is likely to fail without the proper paperwork. Okay, so there's a manifest that details on one row per specimen or per tube for every specimen. And in this manifest, we will, if you decide to join the project, we will send you later the specific details. But I wanted to give you a quick sense of what's involved in this today so that you, so that you know what to expect. The manifest looks like this. There'll be a tab that asks for information on people who are involved at an authorship level on collecting and identifying that specimen or the specimens contained within. And then it's basically go through and fill in this information with you'll get instruction forms of how to do it. The barcode will have a number on it, so it'll be some kind of code here. Specimen ID will come to you later after your manifest has been validated. And then here you're just filling in things like diptera and Culicity. You're going through and filling in each thing. Taxon ID, there'll be instructions on how to fill that in. It's basically you go to the Taxonomy ID website, type in the species name, and a number will come up that you have to enter in here. That means that helps everything stay kind of connected in terms of changing taxonomies. And then the species name, of course. If there's anything specific, like it's a part of a species complex or it's a known colony that you have on site, you can add that information here. And then there's a bunch of drop-down menus. So largely these will be adults, we assume. And then you add in the sort of details of what the nature of the specimen is that's in the tube. So it might be that you've got the whole organism in there. That's what we would ask for in this case. In the past, we've asked for broods. But we don't expect you to go rearing insects. However, if you have an F1, for example, you collected a wild female that was bred and you reared up her offspring, relationship is a place where you would say these are all siblings with each other. This is a place where you put your ID of what you call it, and then all of our dates we expected in this format that it sort of pops up as you hover over it. It needs to be in this year, month, day format. Where it was collected, the GPS coordinates and decimals of where it was collected, and then the habitat it was collected from. These original ones are all about if you're sending in colony material. So if it's colony you've had on site for a long time and you know the original information about where the establishment specimens were from, that would be information you enter here. Otherwise, you don't put anything. And then how you collected the specimen is here. And how you identified it is here, and how you preserved it is here, including, for example, you would want to tell us here, for example, how long you kept it at various temperatures and various preservatives, and what preservative solution it is. And that's basically it, just to give you a sense of what's going to be required. And then each row, we need a row per tube. So once you've filled that in, you send us a link back and then we validate it, which means we check it to make sure it's complete and correct. And and then we tell you, okay, everything's good and we're ready to receive your specimens. There's also some other paperwork around the legal ownership. So either we need an MTA if you or your institute requires MTAs to share material. Or your ABS, which vessel will go into around Nagoya or ABS with sometimes an MTA is required for that. So you may need an MTA because at Sanger we handle so many partnerships and so many specimens coming in. We have a standardized Tree of Life MTA that we're happy for you to use if that works for you. If not, we're happy to accept your legal MTA. We don't think MTAs are always necessary for projects like this because largely everybody's on the same page. We're trying to use the specimens to create a reference genome that will be public. There's no IP. And also, if there's any material remaining, we want to use that or give it to a museum rather than destroy it. And MTAs often have lots of other things in them that are around IP and destruction of materials. So, so just you can consider for your own for each of you whether an MTA is what you need or whether a donation form might work. And a donation form is just a really simple thing that doesn't need to involve the legal department at your institute. It can be signed by you if you are the owner of the specimens and you collected them. And it just says, I'm giving you these specimens. We have an understanding that this will be used for the data generation and that will be publicly released. You will also need to make sure that you have permission to collect. People, you can't go collect anywhere without permission. It's kind of, the permission is obviously very local in terms of how you gain permission. So you will know what to do there as you collect mosquitoes all the time. So it might be that you need local permits or landowner authorization. If the mosquitoes are being collected from any protected areas, any kind of national reserves that are protected, it's quite possible that we will ask for to see a copy of your permissions, just to avoid any downstream illegal collections. So, okay, then this is just to give you a sense of what to expect. From the point at which we receive your samples, so onboarding, that sort of process of getting your samples collected and all the paperwork that needs to be completed, it can take a length of time. It usually takes at least a month, sometimes it can be much longer. Once we actually receive your specimens, we would ask for you to consider that it's likely to take us about a year. The reality is it can take anywhere from a few months to a couple of years, depending on how smoothly things go. But on average, within a year, we expect that the genome should be released if everything has gone smoothly. So as I said, the annotations don't happen until the assembly is released, and these can take some time on top. So this would just be the chromosomal sequence that gets released, and then there will be more time that happens before the actual gene annotations are added on. Once the genome's released, we will most likely draft what we call a genome note, and we have a whole kind of gateway at the Wellcome Open Research venue called Tree of Life, where all of our genome notes go. It's not a scientific kind of analysis of the publication of the data. It's really kind of a mark that says, this is the material we used, this is the quality of the genome, and it's a citation, and it's an announcement that this genome is out there. The genome goes out before the genome note does. So the genome is publicly released as soon as possible. The genome note sometimes takes a little while to put together. Our policy is that the first author of genome notes are always the person who collected the specimen. So you would be first author as the collector. And then there's a bunch of kind of collective authorships of all those teams at the beginning I told you who were involved in all the process. Okay, so to summarize, partner onboarding. If you want to contribute to the project, we will onboard you as a specimen contributing partner to Sanger Tree of Life. We would need the MTA, the donation form, the ABS and Nagoya completion, which Vasu will talk about next. We'll send you a link to the collector onboarding form, and this declares a lot of information. This helps you declare information about like what you intend to send, what materials you might need. For example, if you need the tubes or the barcode stickers or the RNA later or the All Protect, we can send that to you. You would complete the manifest and submit the manifest and get the confirmation that it's validated and it's correct. And then wildlife compliance here would go through that manifest during that process and make sure that the GPS coordinates of your collections are compliant with any kind of wildlife compliance. And then after all of that is done, we would invite you to ship your samples. Okay, so a couple of frequently asked questions. What would we do if you want to put forward a species that actually already has a reference genome? We would still consider generating a reference genome for a species like that if the contig N50 is less than a megabase. So if it's still a relatively discontinuous reference genome. If it's greater than a megabase, we'll take the specimens, but we won't prioritize them. They'll go to the back of the queue, and we will prioritize species that don't have genomes. Many of you have proposed to collect the same species. We're not going to try to say yes to you and no to you. We're going to actually accept all contributions. And the reason for that is that things fall down along the way. So sometimes people don't actually collect what they think they're going to collect. Sometimes the DNA is not the right quality. Sometimes the assembly just doesn't work and we need to try again, and then we've run out of specimens. So we will accept all of your specimens. And we will be as transparent as we can along the way as to what species we've received from whom, so that if you see you're about to go collect in 6 months and you see somebody's already collected 3 of the 5 species you propose to collect, you might decide, well, those are not that, if I come across them, great, but I'm not going to kill myself trying to find them. What if you can't identify your specimens to species level? If you've got a pretty good guess as to what it is, and that guess has ITS or CO1 data out there, we could still consider accepting it with the provision that actually we could generate the barcode data here and make sure that it actually matches what's out in the reference databases. And then if it was, we would put that name on the species and generate the reference genome. As we all know, lots of mosquitoes are complicated in terms of putting a species ID on. And so we will probably have to think about this on a case-by-case basis. If you're really keen to get a species sequence, but you're not entirely sure which species of which complex member it is, for example, I'm sure that we can work around the complex. If you have no idea what it is, then I think that generating a reference genome for it is probably not the best first step. The first thing would be to generate some barcode data for it and see if we can place it. And then finally, what if I have specimens that are really important, but they're not collected to the standard required? For that, I think it's a case-by-case situation. So I would just get in touch with us and tell us how your specimens were collected and stored, and we can discuss if it might be usable. Okay, so that's it from me. I want to thank you all for attending and for being interested in participating. I think it's really exciting to have got so much interest, and I think it goes to show how much people really want reference genomes for the species that they're working on. I'm going to hand over to Vasil now, and I just want to say that with respect to this webinar and the documentation we will send you, feel free to share it with anyone else who you think might be interested in participating in this project. And if you have questions for me in particular, we'll take them at the end after each of us has had a chance to go through. So keep your questions listed, and we'll get to them afterwards. And I'll stop sharing now and hand over to Vasil.
Can you see my screen?
Yes.
Yes, amazing. So, hello everyone. My name is Vasil, and I'm Research Governance Officer in Wellcome Center Institute, and I support Tree of Life program with Nagoya protocol and access and benefits sharing requirements. In my short presentation, in my short slides, I'm going to try to explain the definition of Nagoya protocol and ABS, and also I will try to explain how it could impact to our project. So, what should we know before starting to utilize non-human genetic resources? First of all, we have to understand that what is non-human genetic resources? Non-human genetic resources, it's everything that is non-human and has DNA. It could be bacteria, mosquitoes, plants, etc., etc. And also very important to understand that each country has its own sovereign rights for its genetic resources. It means that each country could establish its own laws and rules how to access to its genetic resources. Saying access, I mean making some research. Just few words about definition, the access and benefit sharing, ABS. I will use this acronym ABS and Nagoya protocol. So what is it? Access and benefits sharing, ABS, it's about how people can get permits to access to genetic resources and how they have to share benefits. So benefits could be monetary or non-monetary. In our cases, we are speaking about non-commercial research and non-commercial purposes. So, and Nagoya protocol. Nagoya protocol is just is international agreement. So Nagoya protocol doesn't create any rules about itself, but it establishes some obligations for countries to provide ABS measures in countries. So key information could be found on ABS Clearinghouse website. But unfortunately, this website doesn't have full information, and but it's quite useful for beginning. Why do we speak about ABS and Nagoya? So it's very important because we are trying to avoid illegal use of genetic resources. It's very, very important for us because illegal use of genetic resources. So if we will use any genetic resources illegal way, there are different fines and penalty in different countries. Country even could exit access to institution for some period or forever. Or also we can have some reputational risks. For our institute, it's very important not to have and avoid any reputational risk. So that is why starting any research activity, we have to understand all ABS rules as internal, on international and national level. As I told you, for each country has its sovereign rights to genetic resources. That is why all requirements in different countries are different, because each country could establish its own laws and its own rules. Just for example, if I have an apple, I can establish any rules how you can get it. I can put it in public domain. It means you can just come to my room and take this apple without anything. Another scenario, I can establish that you have to ask permit, just direct permit. Or unfortunately, there are some examples that there are some tricky rules. And for example, you can take this apple if you come to me on Thursday in the red dress. So yeah, sometimes rules in different countries are very tricky. So I try to split countries for 4 categories. So this is list of countries of country of origin of genetic resources. It's very just very complicated to split to any categories, but I'll try. So first category is the easiest one, the category where there is no any ABS rules. It means you can just come there and take this apple without asking any permits. But it's just from ABS perspective. There could be different legal framework that you have to comply. The second category is countries where there are some ABS requirements, but they are very clear and are not complicated to follow. The third category is category of countries where there are some ABS rules and they are very clear, but procedure is quite complicated and can take some time or additional documents, etc. And the fourth category is countries where ABS rules are not understandable, unfortunately, or they are very complex. So let me give few examples from each category. So even countries from one category could be different, and according to different scenario, one country could jump from category 2, for example, to category for. So, few examples. The United Kingdom. So, United Kingdom is a party to Nagoya Protocol. So, United Kingdom has its own ABS legislation, but it doesn't regulate access to genetic resources for non-commercial purposes. It means the first scenario with free apple in the room. And second example is United States. So it's quite different because United States doesn't have its Nagoya legislation because United States is not a party to Nagoya Protocol. That is why they do not regulate access to genetic resources. But again, they have a lot of wildlife laws. So again, the same scenario of free apple. Second category, I would like to mention Spain. It's very interesting because there are different rules based on purposes of research. For our case, it will be taxonomic purposes. That is why all genetic resources that are used for non-commercial, strictly taxonomic purposes are exempt from legislation. If we will use samples for another purposes, the same samples will be covered by ABS regulation, and we should follow the process. Third category, so I would like to also just quickly mention 2 countries, for example, Brazil. So for this, to bring something from Brazil, we have to have in-country collaborators. So the process says that only in-country institution could registrate in system. So no any foreigners do not have any ability to register in the system. In this case, we definitely need in-country collaborator. That is why this procedure could be in category 3 when we have in-country collaborator and have all process, or can just be category 4 when it's impossible. Very interesting example, Mexico, but I think you will hear about Mexico a little bit later. So, and the last category is category when ABS requirements are not understandable or very complicated. One example of very complicated requirements is Ethiopia. It's complicated process, and even few ministries are involved in this process. So it's took quite a long time and very complicated. And also I would like to mention Tanzania. So we understand, we know that there are some ABS legislation in place, so there are some ABS requirements in place, but we do not understand the procedure. So we try to mail different government institutions to different ministries, but unfortunately, we still the procedure is still not clear. What should you do if you don't know or understand the requirements for accessing genetic resources? So if you will participate in this project, so there could be some scenario when you can't understand what should you do from ABS perspective. What should you do to propose us legal samples? So there is just only one scenario. What shall you do? So first of all, you should check the Google Doc. We will create a Google Doc, and I will try to put as much information as I can there. So you could find there some kind of guidance or some useful information or information how to start the process, but information for each country will be different because, again, of different scenarios. If it doesn't work for you, please feel free to email me directly in any time. Here is my email. So just if you have any questions, please. No any questions? Yeah. Yes, please, Amy.
Yeah, no, so I'm talking from the perspective of Peru, And there is an exportation permit process that we're pretty familiar with, but we always need to name our partners. So like we would need to include some representative from Sanger in our permit process. And so I guess I would just say, sort of, how would we, contact information. I mean, we would need, and then we also would need to include a brief description of the methodologies that you're using. Like, I mean, it's just, they want to know, I mean, down to, that's just something we would have to do from Peru.
Yes.
So
Yeah.
I guess we would just work with you to get that taken care of.
Yeah, so I mean, thank you so much for the question, and it's very nice to hear that you are familiar with the process, ABS process in Peru. It's sometimes it could be a little bit complicated. We have a lot of experience working with Peru, and some of our employers have had a lot of fieldworks in Peru, and bring some butterflies from there. Answering your question, so yes, you have to give them information about your partner. So as my understanding is the partner will be Welcome Sana Institute, just institution, and PI will be Mara. And about methodology, I think Mara could explain, because I don't know anything about methodology and etc.
That's great. It's not, I mean, it's tedious. We're in category 3, I think. But again, we have a lot of experience shipping samples out, but that's something I know will be a requirement for us.
Yes, I just maybe short disclaimer. This table is very approximate. I tried to split, but yeah. So if you know the process to bring samples from Peru, I think we can do it without any problems. But I have had some experience when person do not know the requirements of Peru, and it took some time. But yeah, everything is manageable, and as I told you, we have some samples from Peru. Have had. So.
And Amy, I think in terms of protocols, we can upload a generic description of the protocols that could be used.
That's exactly what we need. And we just need,
Yeah.
like I said, who would be the contact person, and then like a shipping, like the shipping address, literally. I know it's like very high, very fine scale details, but we need to have that information up. And it will take 3 or 4 months maybe to get the initial permit, and then you have to do export permits aside from that. So it takes some time.
Absolutely. Yeah, we can upload onto our groups that we'll talk about more later, those kind of documents that can be used across different contributors.
Shall we move on to you, Miguel, and then take any further questions for Vasil at the end? You've got to unmute, Miguel.
Sorry. Thank you so much, Vasil, for your description. I'll give an example from Mexico. Can everyone see my slides? Yeah. Okay, so as I mentioned, I'm at the National University of Mexico, the UNAM. And so I'll give an example of how I've been working with different contributors from Mexico to try and create a single hub to be able to submit specimens for the project. So Nagoya and ABS of the identification of national focal points and competent national authorities, which are the kind of jargon that's used within the accords. These are within the Secretaría de Medio Ambiente y Recursos Naturales, which is a government ministry, a federal government ministry, which regulates Nagoya Protocol. And I guess luckily, within my university, an institute that's separate from my own, I'm at the Institute for Biomedical Science, but the Institute of Biology has or houses the National Collection of Insects. And so they have experience and president for exporting specimens from the national collection to collaborators. And they have the collection permits and export permits to be able to do this. So we've generated, together with the School of Veterinary Sciences, which is another school, institute within the UNAM, to generate an intra-institution agreement that will enable us to use the permits through the National Collection of Insects to carry out exports of some of the mosquito species. We're also working with some of the partners who have answered this call within Mexico. These include Vicky Bando in the National Institute of Public Health and collaborators at the Autonomous University of Nuevo León, who we will be working with to try and generate inter-institution agreements, so outside of the UNAM, with these other national institutes to be able to channel some of the exports, hopefully through the UNAM and the agreements they have with the Semarnat. So this is kind of the framework of a national hub, and perhaps teams that are within countries can consider something similar. These are some of the numbers that have been of the species that have been proposed by different contributors who've answered the call so far. So in total, we have a total of 153 unique species, many from Aedes genus and Culex and Anopheles, but a number of smaller number of other genus that are less well represented in understudied, let's say, in general in the field, but some interesting diversity from different parts of the world. And as I mentioned, different countries, these numbers on top of each bar are the number of contributors from each country. So within some of these countries, it may be something to consider to generate one of these hubs to minimize the amount of work that's done and channel some of the exporting through a particular hub or institution that wants to take on that role. So in Mexico, we're doing, as I mentioned, that with the 3 institutions that form part of the contributors. And so I'll just leave these up so you can see the number of contributors from each country so far. And then finally, this is just a list of those species that have been proposed. These are species that are unique. So contributors, if you see the species that you proposed here, it means you're the only person who has that collection currently that's being proposed, and it would be, very interesting to be able to generate those genomes. And then I'll just leave this up for a couple of seconds so you can see, but of course we'll have this in the recording. And then the next slide is just the species that have been proposed by multiple contributors and how many contributors are the numbers. And so these are perhaps more commonly found in different geographies. And as Mara mentioned, we're still interested in receiving those because of numerous logistical issues along the way. So I just wanted to put these up so people can see what's being proposed so far. And I'll stop there for now, Mara, and take questions later on.
Thanks, Miguel. And then our final speaker before Q&A session is Bernard.
Okay. All right, so I just wanted to wrap this up by talking about not just generating genomes, but what we kind of envision what these genomes will help the entire field do? And I think this is a, isn't that going? I think this is a, kind of pretty common question for any of these large biodiversity projects is, we're generating thousands of genomes, but, what are we going to be able to do with them? What new insights are we going to be able to generate? So, I think it's, clear that this project is just one of many efforts to sequence biodiversity across much broader swaths of life than just insect families. So kind of in the near term, we, as Mara said, we expect to have genome assemblies, transcriptome data, and maybe some short and long read resequencing. But, I think to make this more generally usable, there are some next steps like genome annotations, both gene annotations, TE annotations, and other kinds of genome annotations. There are whole genome alignments, genomes, orthology group inference, D and DS calculations, alignments, PopGen data sets, and I think Mara's group has been working on this in particular for mosquitoes, but all of this can also be used as a resource for people working in the field, sampling new species and figuring out what they're collecting actually is. And so, I think we, are hoping to sort of help establish working groups for doing a lot of these things to, both standardize methods used across the field, but also to kind of encourage collaboration because we're generating this huge resource that, there are so many things that you can do with it. And I think it's also important to highlight when we're working with this scale of data, it's really important to do this in a scalable and reproducible way. And so, any of this that we do in the future, we hope that, it's going to be done with standardized pipelines, that people know exactly what's going on under the hood, containerized workflows, so that even the versions of computer programs that are being used to generate each one of these things is sort of consistent and everyone kind of knows what's going on. Sometimes you can have a program, the version changes, and then you end up with slightly or drastically different results. And of course, trying to accelerate this with the latest in computing tools like, GPU driven workflows for doing genomics. And so, of course, all of this is still very much early days. And as Mara mentioned earlier too, the sort of limiting step to even having the data to do all of this is by far field collection. And so, this is where we are hoping that people like you will be interested and, help contribute samples so we can build all of these resources together. In order to do this, we've tried to set up a Google Groups to maybe facilitate some of this discussion. So I'll put this link up again at the end, and we'll send this out through the email. But we're set up a Google group. Hopefully people can ask questions there so that, I'm sure there will be frequently asked questions, and it's hopefully is somewhere where people can go and just search answers to their questions as well as ask new questions too. And we're hoping to kind of integrate this with the Google Doc and other resources that we have. Okay, so just to leave everyone with a small final thought, I think again, we having a lot of genomes is nice, but what are the things that we can do that we couldn't do before with this kind of very clade focused sampling? And one of the questions I'm really interested in is trying to understand, kind of like every other evolutionary genomicist, how natural selection works and how it's connected to biological function. And so population genomic data are in principle the best way to understand and estimate natural selection. But even in high diversity species like insects, and I work in Drosophila, so in Drosophila melanogaster, you have very little polymorphism across the genome. And just as an example, here's an enzyme. It doesn't really matter what the enzyme is, except that it has a structure and has some catalytic residues highlighted here, also shown in linear space through these annotations. And if you take the population genomic data from a single species, you have 10 SNPs, which really just illustrates that, because there's so little data, or because it's so sparse, you can't really learn anything about the function of this protein through population genomic data from a single species. And this has kind of been a major challenge for my field for, the last however many years. And so, what we are doing in Drosophila and what we hope to, do something similar for this mosquito data set, we have sequenced, over 550 genomes of different Drosophilid species. So, in terms of the total evolutionary time that encompasses, that's billions of years cumulatively. And, we get hundreds of expected substitutions per neutral site. And we've started to add population genomic data around the tree. And right now, that's much sparser, but with 150 species, we have over 5 SNPs per neutral codon. So what that gives us really is going from a single species, you get to, when you start doing 15 or 40 to even 150, you achieve saturation of variation across every possible site in the genome. Of course, this is aligned across a lot of different species, and there's caveats to that. But what that really lets us do is, if we're interested in these questions about evolutionary rates and function, we can take an enzyme like this, like that I just showed you, and overall, it's a fairly conserved enzyme. DNDS is very low, and we can see what those patterns are over the entirety of the enzyme. And clearly, the single number doesn't describe everything. There's a lot of variation, and what you can kind of start to see is that the variation in evolutionary rates is very closely related to the function of the enzyme. So right around the catalytic residues, you have very, very strong constraint. Every species is the same, and there's no divergence at all. But overall, this enzyme, it's fairly constrained, and you wouldn't really say from this kind of data that there's any adaptation happening in this enzyme. But when you layer population genomic data across this, you see consistency with strong constraint around the most functional parts of the enzyme. But you also see that there are other portions of the enzyme where there's no variation across any species. And we know that this isn't due to chance or the sparsity of variants because we have saturation. We know that this is because there's strong selection in every species, yet somehow changes are occurring across different species. So, we think that's kind of the clearest signal of adaptation we have, and that you couldn't really see without having this kind of very, very focused clade scale sampling. And what's quite cool about this too is that if you kind of think about function itself, it's a 3D, something that's happening in 3D space and not over kind of linear genome. And the strongest constraint actually does cluster spatially. And you do see signals of adaptation, but they never happen exactly where the most important function is. They happen at places that can impact the function, and tune it, but not break the enzyme itself. So I mean, this is just one vignette of things that we're interested in doing, but this is something that we've kind of just come to being able to do for Drosophila. And it'd be fascinating to see, what this looks like in mosquitoes if we have the same genes and the same proteins. Do we see similarities? And do we see shifts, and how do those shifts occur across these proteins, and how much of it might be related to particular, ecological or macroevolutionary factors? So yeah, hopefully that's interesting. And again, here's the link to the Google Groups. The QR code leads to it, and we will send out the link. And we hope that people will participate, and that this will facilitate collaborations and future science in the system. Thanks.
Thanks, Bernard. Okay, so I think that concludes everything we wanted to say. I'll just add on to what Bernard just presented, which is, our ambition with the reference genome project is to just use the minimal number of samples that you send us to get those reference genomes. So all the extras that don't get used up in the process of reference genome creation could theoretically be used for short read sequencing for some of those PopGen, so starting to explore within species diversity. And I think the other thing which we mentioned in our Google survey that you all filled in, Google form that you filled in, is that this is kind of a pilot. This is really us trying to get to grips with being kind of on that cusp of being able to really do this at scale. And we would like to go in at some point in the next probably 2 to 3 years with a pitch to do the, to do the full family. But we're starting with 100 species now. So it is, I think, be thinking about that when you're collecting, even if you're not interested in participating now, if you might be interested in participating in the future. I think the project is likely to go on for some time. And I think now, if you have any other questions from anything any of us presented, the floor is over to you. Masamba?
Okay, good morning, everybody. Thank you. That was really clear. I have some concern because here, for example, in Senegal, if you want to collect Iranian Mayri, Ludzia, or Toxaranchitis, if we really need fresh and good specimens, we have to travel to the southeastern part of Senegal during a deep rainy season to get really fresh, good samples to work with. Do you think that it won't be really better to have at least some basic financial support to organize kind of field trip just for collecting those specific samples? This is my first concern. For example, we can try with Wellcome Trust, NIH, Bill Gates, to do really a good focus on what we want to do. Another observation that I have is that I quickly run through the list of species that were targeted, and I saw Aedes africanus, I saw Aedes neoafricanus, which is from the Africanus group, but there was no mention of Aedes cornetti, which pose a real problem of systematic, because differentiating Africanus from Neo-Africanus is not a big deal. But if you integrate the third species of the Africanus group, which differ from the Neo-Africanus one, just by 2 dots on the legs, the third legs, I think doing some genomic of these species will be really helpful, resolving some status of specimen that still exists. Okay, so if we need accurate identification also, we need really fresh specimen. For example, if you use CDC light traps overnight, you can get some specimens, but the time you get those mosquitoes, most of the features are rubbed off, and you are not really certain of what you are telling. So I mean, we must go from a typical taxonomic status of any single mosquito before going to do some deep genomic work. I mean, the key approach is an accurate identification of specimens. So if you want to do that, we need really fresh specimen also. That's why my idea of finding at least basic support, for getting those species that are really a little bit hard to collect. It asks some particular efforts to collect.
Yes.
Getting those funding will be a really good thing.
Thank you, Masamba.
Yeah.
I mean, I think you're completely correct on both accounts, and we would love to be able to financially support collections, but we don't have that kind of budget now. So I think that if we think of this in a 2-phase approach, phase 1, this pilot that we're trying to do now, is the stuff that is easier. It's the things that people come across anyway, and that they can identify, more straightforward with fresh collections. They can get fresh collections more easily. It sounds like for the species that you're interested, that you would need a special trip for. It might be that would be a better wait till we try to seek funding. When we seek funding, absolutely funding will be in there for collections and for identifications, for the additional work that's needed to do identifications. At the moment, that's not, we're doing a pilot, right? We're doing kind of a demo of what's possible on the technical side, trying to take advantage of people doing collections anyway, who want reference genomes for those species anyway. There's obviously much more to be done. And I think it's really your decision as to whether this is something that's feasible for you to do now, or whether you would like to stay in touch and kind of, or maybe submit some samples from species that you come across easily, and leave the harder ones for the later phases and try to seek some funding collaboratively with this group at that stage. Amy?
I sort of following on that, what sort of like taxonomic metadata do you want, or validation? Do you need like photos? Do you need like a brief, like run a key? What sort of information do you want collected on the taxonomic side, for future validation, I guess. And then I also had a question about ironically, it's kind of hard to get 100% ethanol where I am in Iquitos, and it's a controlled substance in Peru, so it sounds weird, but it's not necessarily easy to get our hands on. And I would just want to say we've developed a protocol with DNA Shield with Aedes aegypti, where we've been able to get high quality DNA extracted, and I would be interested if you are interested in entertaining some alternative protocols. So
yeah, thanks. So for sure, a photo would be fantastic. We trust that everybody who's participating in this project and contributing specimens is a taxonomic expert, and with the caveats that we put forward, saying we really need it to be confidently ID'd by an expert. As you know, to the best of our abilities, we know how challenging mosquitoes are, and things do go wrong.
I mean, I do work with someone who's really good, but I mean, anybody who does taxonomy and you need to work quickly, I think knows that, there's a certain degree of error sometimes. Yeah. And yeah, sort of just, if there's any characteristics, I mean, we'll definitely try to take photos if possible. And
on DNA Shield, we actually have tests, I think, in the next month to see how well that preserves high molecular weight DNA. So we know it's really good for inactivating viruses. We think it would be a really great, it also protects RNA, if I understand correctly. And
no, that's exactly what we've done, is we pull both.
Yeah. And
I could put you in touch with the person who sort of developed those protocols for us, if you like.
Yeah, that would be great. The one challenge with that on our side is that it has to go through a specific extraction protocol then that's not our typical high molecular weight extraction protocol, but we haven't yet tested to see how good the DNA is in terms of long read sequencing. It's probably going to be okay. So I would say, we can hedge our bets specifically with you on this and decide, whether maybe we ask you to put some specimens in 80% ethanol, or something that's like, just below the threshold of what's controlled, plus some in DNA Shield, and then we see how it works.
Sure. No, that's, yeah, I'll have to be in touch anyway for permits. So
yeah. Okay, great. Thank you.
Okay.
Andreas?
Yeah, thank you, Mara. So my question is still on the identification. It's very difficult for some of the curious species we find here to be identified to the species level. So in this case, how do you target a species, or you can target a genus?
So we don't think we can target just a genus at this stage. I think it would be good. We should develop processes in this pilot to handle the things that are difficult to identify. So I think what I would suggest for those cases is that we work directly with you to talk about how you would provide us specimens that are identified only to genus level. And then on our side, we do a good extraction that would give us good DNA, but that we first do some additional barcode sequencing to see how well we can classify and cluster the different specimens into species-like entities. And then we discuss with you and with other experts on that genus how to take that forward. I think that that's an important part of this pilot, but it's, in my mind, it sits in a different level than the sort of straightforward ones. And that's the majority of what we want to try to do this pilot is generate, 100 high quality genomes for named species that have names. Alongside that, we want to figure out how do you handle the things that you can't identify morphologically to a species level, and even molecularly might be tricky to identify to a species level, but still we want reference genome data, resources for these organisms. So I think my answer to you is a little fuzzy. I would say get in touch with us, I think, about exactly how we would approach something that where you're pretty confident on the genus but can't get it to species level, what we might ask you to do and provide in that case.
Yeah, thank you.
Thank you. Ryan?
Hey, Mara and others. So I had more of a technical question. So I oversee a surveillance program. And so some of your mentionings of permissions and authorship kind of, they get a little fuzzy. And I wasn't sure quite how to potentially deal with that. So oftentimes we have partners in local areas that we're not doing the direct collections. I guess what level of involvement would you suggest there? We do not have any kind of written formal permissions oftentimes. It's local partners that are running these oftentimes in public areas. Those are the types of questions that I had, if you can kind of comment on.
So on the permissions one, I would say that it's probably a longer conversation that we would have with some of the teams here on exactly what, you almost certainly have permission to set up traps and to collect what you're collecting. And I think that that's probably all we would want to know. On the authorship one, it's sort of your call. I think it is complicated when you have teams and it's lots of people and you don't know exactly which person handled which insect. In which case, it's, I think, kind of up to you how you might do a collective authorship approach, sort of say this is this, sort of surveillance team and they were all participating in all these species and then they're all named. That's kind of how we approach it when we don't really know who did exactly what on which species here. And it gives credit to everybody still.
Okay, great.
But happy to continue those conversations with you. And if anybody on Toll's side wants to add anything, Nancy or no. Yeah, okay.
Yeah, and then to kind of follow up with a second question, would you recommend that maybe those of us in the United States get together and kind of coordinate samples, I guess, to some extent, and how you recommend us doing that? Is that through that Google chat or I see a couple familiar names here. Maybe we can kind of work together.
Yeah, I think there were a lot of species. US was, I think, the top, in terms of numbers of species and numbers of people who are proposing species, was in the top couple. So I think that's part of why Miguel did that demo is to sort of get across the value of potentially having a hub. And the US does seem like a really sensible place to have a hub. And for you guys to coordinate a little bit on, especially where there's complexities about finding certain species or whether there's species complexes, and figuring out who might have. So I think the hub concept is one that's being developed now a little bit more in a refined way on our side around, I think you would still, if you were the hub and everybody was sending samples to you, you would still need to make sure that you had some responsibility about kind of they had permission to collect and actually the US in some respects is the place that's probably in terms of ABS / Nagoya the least needed for a hub because actually there is no ABS requirement there. You can send your samples to us without going through that. You still need to do all the other things, the MTA / donation form, the permissions to collect, the manifest, etc. So I think it's probably worth another conversation around like the pros and cons of a hub in the US. In terms of replication, it certainly makes sense for you guys to post on the Google groups, and we'll figure out some way to make sure like it's transparent who's really intending to submit which species so that people are aware of what's coming, and whether to coordinate some of that within US.
Okay, great. Thank you.
Evelyn?
Yeah, thanks a lot, Mara. Can you hear me?
Yes.
Yeah. So my question is around cost and specifically the cost of shipping the 10 or 20 samples of individual species of mosquitoes. And I ask that because I support a couple of national malaria control programs within East, Southern, Western Africa, and in LAC as well, Latin America. So I'm just wondering, when it comes to cost, is there an average price for shipping that we are looking at? Because on my end, I'll definitely
It does
do some
depend a lot on where you are. I think for an overnight or kind of a short-term room temperature envelope containing, 30 or 40 tubes, it should be on the order of, I would guess, $20 to $50 per shipment. Nancy, do you want to add any additional considerations there?
No, I don't think so. If it's being shipped ambiently, that price sounds about right. There probably is some variability depending on where it's coming from.
Yeah, we've had dry ice shipments from Cameroon cost us like £3,000, which is more than the price of the genome. So this is why we're really trying to move towards room temperature shipment. Consideration for the hubs, though, which is if anybody does form a hub, the cost of shipping a dry shipper is not as high as trying to do dry ice. And so it is possible you could collect a lot of different specimens and do one very large shipment in a dry shipper and keep everything at cold storage. I think I would still advise to put things in the preservatives just because it's a good backup in case things go wrong.
Okay, thanks, Mara.
Thanks, Evelyn, for your question. I don't see any other questions. Oh, Maria, go ahead.
Hi, Mara. I'm Maria Sharakova from Virginia Tech in the US. And in my lab, we are specifically interested in chromosomal rearrangements, studying chromosomal rearrangements. And we are planning to do high-C on the specimen, which we will have to see how much they rearranged. And could you just explain a little bit more clearly what are your quality expectations? So do you plan to assemble all at chromosomal level? All assemblies will be at chromosomal level. If you're interested in collaboration, let's say we will do high-C and share this data.
Yeah, if you're doing high-C on the species anyway, it would be great to know because then we won't do it. But if, yes, we are trying to do chromosomal for everything, so everything will go through high-C.
But actually, could you say something about high-C? How you exactly want to do high-C? Just from one like a single mosquito?
One single mosquito. So that works well enough for just doing the config order and orientation. It's and actually we've done it successfully from just one part of a mosquito. We've used the head for high-C and the rest of the body for the high molecular weight DNA. You don't need to pool them for high-C, but I think for some of the, to get a sense of rearrangements across, within a species, across individuals, that's not data we would be generating because we wouldn't pool individuals for high-C, so we wouldn't get that sense of the variation in
because we usually do high-C for 60 males, usually to see what's going on in the population.
Yeah.
Another thing, we also want to do karyotyping. I don't know if this can be kind of important because sometimes, based on my experience with assemblies, sometimes after you do high-C, you cannot really orient, especially like arms correctly, or even arms can be misconnected. So this is also we want to do with this like a species that we will be working on.
Yeah, so maybe we talk offline about what you might need for karyotyping and whether there'd be any sense in trying to get, because you would need more than one specimen and you would need them preserved in some particular.
For karyotyping, we need the larval stage because we use imaginal discs from larval stage.
Yeah.
And we can do, they can be just preserved in Carnoy solution is ethanol, acetic acid, 3:1.
Yeah.
Then we can use them for karyotyping. So we need probably 10 larvae will be enough.
Yeah. So I don't have a sense of how many people will be doing larvae collections, but we'll try to get a sense of that too. That will be collected in the manifest. And if people are putting forward a lot of larvae, ideally, I think ideally we would like adults. I think we have not done a lot of testing on larvae. So we just are much more familiar with adults. But yeah, let's see what the sort of general framework is in terms of what people are submitting. If they tend to be doing large larval collections and submitting larvae, then it would make sense to maybe ship out some carnoys and then get those samples to you. I think because we'll likely be shipping out the barcoded tubes anyway, so it would be straightforward to ship out some with carnoys in it.
Okay. And also just a little question, because I see that in in Russia you have also several groups, or it's just one group. Who contacted you from
I
Russia?
don't know offhand. Miguel might know.
Just to make sure that we are not duplicating also our efforts.
Sorry, I think it was someone with multiple countries in the list that included Russia. So I'm not sure if they're there. But I can look that up if we want to continue. I'll put the information in the chat. I'll look this up.
I see a question from Alan.
Oh, hi, Mara. Yeah, just a quick question. It's about this presentation's been recorded. I'd like to share it with some colleagues. Will it be sent or is it stored somewhere else?
Yeah, we'll send it out in a follow-up link summarizing kind of what we've covered and a link to the presentation and other documents later this week, probably. And you're welcome to send it to anybody.
Brilliant. Thanks, Mara. Okay, thank you. That was it.
Mara, I think there's a question in the chat.
Yeah, I'm just reading it. So I'm currently collecting different mosquito species from northern Ethiopia. I have a budget limitation to facilitate the shipping process. Yes, so if you don't have budget to ship, we can do reverse charging. It adds a little bit of complexity, but it is possible. So what you'll need to do is find out who your shipping providers are locally, whether you have both FedEx and DHL or something else, and we can try to figure out how to reverse charge the shipping. And then another question, when can we start receiving samples? Well, as was in the presentation, we can start receiving samples as soon as your partner onboarding process is completed. So all of those things that I went through around the metadata manifest, the collections, the barcoded tubes, the making sure that the specimens were alive, the MTA or donation form, the ABS compliance, the permission to collect, all of those things have to be completed before we would receive samples. So we hope that some of you will choose to continue to work with us, and that we'll send you some information on how, you're not beholden to the species list you put in. That was really our initial taking a pulse of the field and trying to understand whether we could get enough interest across all of you to get to 100 species, and it looks like that's possible. So I think we want to move forward with this pilot. So now it's about really getting a lot of you through that process, because it is a time-consuming process in addition to all the collections that have to be done, which are time-consuming and expensive. So I think what we'll do is just see how many of you actually decide to participate and kind of join the project and get through those steps. So to answer the question, there's really no date. We can start receiving samples tomorrow, but we can't receive any samples until partners are all the way through that process. Thank you all very much. Are there any final questions? We're coming up to the half hour here. I don't see any more hands.
Just to confirm, Maria, the only group from Russia was yours proposing USA and Russia. Yeah.
Okay. Thank you all so much for sticking with us for the hour and a half. And we will, and thanks to all the presenters and for the toll teams and singer teams to being on call. We'll be in touch by email to all of you and give you all the links to join the Google groups and to begin the process of partner onboarding and telling us what you what you need help with and what you need in terms of supplies. And yeah, thanks to everyone. Does anybody?
Are you sharing the presentations or not?
Well, we'll share the link to the recording. If you also want a link to the actual slides, let us know and we can send them to you.
Okay.
Anything from Miguel or Bernard or
I'll place the list of species up on the Google Groups link as well, and my slides. Yeah.
Oh, that's a good idea. Yeah, we can put our slides on the Google Groups. Yeah. Okay.
Yeah, nothing for me. I'm excited.
Okay, thank you all so much for joining and for your interest in the project, and we hope you will remain interested. Take care. Bye-bye.
Okay, bye everyone.
Thank you.
Bye.
Bye bye.